Good morning. I hope everybody's wide awake. We're going to change gears a little bit. I'm actually in awe of all the work that you folks do on the ground and the good that you're doing in the world for dogs. And this talk is a little bit of a change of pace. I'm going to tell you about hopefully some tools that my industry might be able to provide you folks to make your work more effective. So we'll jump right in, and I'm going to tell you briefly about what the talk is going to be about. I'm going to tell you my philosophy about a toolbox approach to developing sterilants and contraceptives. I'm going to talk about what's available now, which is actually fairly limited. I'm going to touch on what regulatory approval means in my world so that it will help you understand what your choices are. And then I'm going to give you an overview of some really jazzy, exciting new research that's happening in our field. Touch briefly on barriers and issues, so as not to be too depressing. And then talk a little bit about work that's being done to support the use of non-surgical contraceptives. And finally, I'm going to talk about the question that's on everybody's mind, which is when are we going to have these things and when will they be available? So I thought it would be fun to go back in the literature and pick out the first publications on non-surgical contraception for the use in dogs. And as you look at these, you'll see that there's been publications as early as 1975 on using a immunocontraception. But as of yet, we have no product that's effectively can be used for dogs. The second immunocontraceptive approach was using zoonopelucida vaccines, and although we have these kinds of vaccines for contraception in horses and wildlife, but yet no dog product. And the last one is one we actually do have a product. It's the first publication in 1984 using a GnRH agonist, and I'll talk more about what that is. And it took about 20 years from that publication to actually get a product that was approved for use. So as you can see, it's a long road from research, first published research, to an actual product. So let's talk a little bit about this idea of a toolbox. Many years ago when I went to my first population management conference, everyone was talking about the silver bullet. We need something that's 100% safe, that works in every case, and that is, I originally put cheap on the slide, but they asked me to put inexpensive instead. But actually the term was cheap. We want something cheap, it works all the time, and there's no risk. And of course there's no product like that. There's no product in human health, and there's no product in veterinary medicine. And I started to think about what this term inexpensive or cheap means, and it's really, in my mind, it's compared to what? And I don't think we really understand the cost of what we're doing now to sterilize animals. So there has to be a comparison made there between something that's pennies per dose and what you're actually spending now to surgically sterilize an animal. And then everything should be looked at in terms of risk and benefit. There are risks to everything that you do, including surgery, and there are benefits to the things that you do. And so as we discuss all these available options that are either available now or going to become available, they all have their drawbacks, but they all have their benefits. And so I like the idea of thinking about developing a whole variety of tools that can work in all of the situations that you folks are working in, and they're not all the same. And a tool that might work for in one situation may not be useful in another. So the idea of having one silver bullet is now replaced by the idea of developing a toolbox. Not to get too deep into physiology, but I'm going to be talking about targets for contraception, and I just wanted to use this slide to point out that there's really three general targets. One is the brain. You can go after the specific neurons in the brain that are the master neurons that regulate reproduction, and these make a hormone called gonadotropin-releasing hormone, and I'm going to abbreviate that by calling it GnRH. You can try to knock that out. You can also work at the level of the pituitary, where GnRH acts to knock out some of the pituitary hormones that control reproduction. And then finally, you can work at the level of the gonads, where you can try to affect the development of ova in the ovary, for example, or the maturation of sperm. So this gives us a wide range of targets that we can look at for potential contraception. And each one of these targets has its risks and its benefits. And I'll talk a little bit about all of those. But let's go back to the toolbox for a minute. And I use this slide to illustrate the variety of things that you might consider as potential tools in your box. You might have an approach, for example, that works on females, that suppresses hormones, and is permanent. And that would be a really impressive tool. Or you might have something that works on males. It suppresses hormones, but it only lasts for 6 months. In some hands, that might be a useful tool. So you can kind of mix and match all of these different things that you're looking at, whether it's surgery, males and females, hormone suppression or hormone maintenance, a wide variety of ways you can get the product into an animal. And whether it's permanent, it's long-term, or it's short-term may have an effect on where you use it, and obviously what species it might work in. So there's a range of possible tools that can mix and match these different attributes. So I'm going to be talking about some products that have regulatory approval and some products that do not and are more experimental. And so I wanted to just briefly explain to you what in my world of drug development regulatory approval means. It basically means 3 things for a product that's going to be used in dogs. It means that there's quality manufacturing that's done under what's called GMP, good manufacturing practices. It means that the product has proved to be safe and proved to be effective. And then there's also this concept of pharmacovigilance, and I put it under safety, but it's a term that means that the company that is selling the product is required by law to provide a way for people to report adverse events. So if you use the product in a dog and it causes the dog to vomit, for example, you have a phone number on the package that you can call, and that's somebody in the company who has to record that your dog vomited and has to report that to the government. And the same thing is true with effectiveness. If the product says it's going to be suppress fertility for 6 months and your dog comes into heat in 2 months, you can call up the company and they're required to report that to the government. And if they get enough complaints that they're getting side effects and lack of effectiveness, the government can take that regulatory approval away. So it's a nice way to hold companies accountable for the safety and effectiveness of their products. You generate all these data to support your claim and you submit it, you get a regulatory approval, but that approval is for that particular species and that indication. So the approval might say for use in dogs to suppress fertility, use in male dogs to suppress fertility for 6 months. If you use it in female dogs, it's considered off-label, but it has a regulatory approval. So you know it's been manufactured in the appropriate way, but you're using it off-label, and that's okay in some places too. It's legal to use some drugs off-label. So that's what regulatory approval means. So when you buy a drug that has that regulatory number on it, you know that the manufacturing has been done in a quality way, and you know that there are data, significant data, that have been reviewed that prove that it's safe and effective. So let's talk about some of the products that have regulatory approval that are available now. In female dogs, I'll talk about one progestational drug, which has a couple of different names in different parts of the world. And then for male dogs, we'll talk about Supralauren and Zuderin. So the 2 progestational drugs have been used they're approved, for example, in the UK and Australia. They're given by injection. You probably are familiar with them. They suppress Asteris for a short period of time. And they're not really very effective tools for dog population management. They're effective tools for someone who doesn't want their bitch to come into heat in the next couple of months because she's going to a show, for example. They have a very narrow safety margin, and so the risk-benefit is, in some cases, not considered worth using these compounds because there's a lot of side effects associated with them. But if they're used responsibly, they can be effective for short term. And they're marketed in Europe and Australia by MSD, Animal Health. Supralauren is a GnRH agonist, and this works at the level of the pituitary, so it actually suppresses GnRH over long periods of time. And it has regulatory approval in Europe and Australia and New Zealand, not in the US. And there's 2 versions of this. It's an implant that suppresses fertility in male dogs for either 6 or 12 months. You give a dog one implant and you don't need to remove that implant. The implant it's a fairly small implant and it kind of melts under the skin, so you don't really even feel it. And it takes about 4 to 6 weeks to suppress fertility, but once fertility is suppressed, it will be suppressed for that 12-month period if you've used the 12-month implant. It's a variable when those dogs will come back to fertility after the implant wears off, and some dogs it lasts a couple of years, and some dogs that come right back into having viable sperm. And this implant is marketed by Virbac. I thought you'd get a kick out of seeing how they advertise it. They like to emphasize that you don't have to cut the testicles off these dogs. Zeuterin is another option. This is also an option where you don't have to surgically remove the testicles. It's called Asteris in some markets. It's a chemical sterilant, zinc gluconate, which is buffered with arginine. It used to be called Nutrasol. They changed the name when they relaunched the product recently. It has regulatory approval in multiple countries, including here in Turkey. It's an intratesticular injection, and I just like to emphasize that it doesn't completely suppress testosterone. There's a lot of controversy about exactly how much testosterone is suppressed, but it's between 40 and 60%. So you get complete permanent sterility, but not complete suppression of testosterone. And this is marketed by a company called Arc Sciences. They're targeting it for sale in shelters, and one of the things they're doing that I think is really makes a lot of sense is they're doing extensive training programs because the technique by which you inject it is very important to minimize side effects. It's a sterile product. Once you open it, you have to use it all, but it comes sterile and it's manufactured under GMP manufacturing. As we talked about in one of the presentations yesterday, when you do a non-surgical sterilization, you need to mark the animals to make them able to be identified as sterile. And the Zeuterin folks advocate using a Zeuterin tattoo, so a little, like the little Zorro Z gets tattooed on the dog's tummy when he gets an injection, and that's an important part of the procedure. So that's it. Those are our regulatory approved contraceptives that are available. There are experimental approaches that have been field tested, and I'm going to break these into two different categories. One is products that have regulatory approval that are used off-label, and the other are products that are used without regulatory approval. Supralauren for female dogs, off-label, and I'll introduce a compound called Gonacon that is being explored off-label for female dogs. In a program called Dogs with No Names, which some of you might have heard about, Dr. Judith Sampson-French uses Supralauren 12-month implants to suppress estrus for 6 to 12 months in bitches in First Nation reserves in Canada. There's a pretty limited data on safety of Supralauren in female dogs, and there are some indications that there might be some safety concerns, particularly in causing endometrial problems. But we know for sure that if you give these implants at a certain stage of estrus, a certain stage of the cycle, you can induce estrus. So you give the implant, you're supposed to be suppressing reproduction, but you actually cause a fertile estrus, which is obviously something we don't want to do. However, in her situation with these dogs that are incredibly difficult to catch, that are on the edges of the community, who have litters of puppies that freeze to death every winter, she's evaluated this as a reasonable risk for these animals. But again, you really, in every situation, you have to do a pretty careful evaluation of the risk versus the benefits, and each situation is different. Some of you might have heard of this vaccine, Gonacon. It's a vaccine against GnRH. It was developed by the National Wildlife Research Center to control populations of wild deer and wild horses, and it has a long duration. It's a kind of a unique vaccine that doesn't need a booster. It's a single injection. It took them over 15 years to get regulatory approval for this. It was a long and complicated development program. And people got very excited about the opportunity of using this in dogs. Unfortunately, the deer formulation causes pretty severe injection site reactions in dogs, and we saw this in a study in Nepal in 2013. So the NWRC is developing a reformulated version, and they are testing it now in dogs. And if it works, it would be a nice way to give a single injection and get suppression over a longer period of time. But we yet don't have a balance of injection site reactions and efficacy that's really acceptable. So this is not really available, and it's under experimental use. Intratesticular calcium chloride, I'm sure some of you have heard of this, some of you may have been using this. It's been in extensive experimental field studies in the US, Italy, and recently there were a couple of publications from India that indicate that it's effective. There's a bit of safety data, not extensive. The calcium chloride is unregulated, so it's not approved, so you have to mix it up. You have to formulate it yourself with alcohol and sterilize it through a filter sterilisation, so you don't have the assurance of GMP manufacturing. But people are using it and advocating its use. You have to carefully evaluate the risk and the benefit of this approach, and each situation is different. But that's it. So we really need more tools, and there really is a feeling that in the past there just hasn't really been much research money available to help us get to more tools. So when you look at human biomedical science, there's so many cool and exciting new breakthroughs happening. Why aren't those breakthroughs coming to help us for our cause? And this was the genesis of Dr. Gary Michelson starting the Michelson Prize and Grants program, which we've talked about previously. He's put $50 million on the table for research into non-surgical contraception and sterilants for dogs and cats. They have funded over 30 projects in 7 countries since they started in 2009. I serve on the scientific advisory board, so I get to see all these grants, and really world-class laboratories are submitting grants for this money. This is Dr. Michelson's goal. He's focused on the US, but he has clearly told us that if products are developed with his funding, that they will be made available for international use. And he's very dedicated to actually bringing these products to market. So he set a very high goal for a prize. If you invent a sterilant that meets all these criteria, you will win $25 million. Believe me, it's not easy. Nobody's won the prize yet. He set a very high bar. He wants permanent, single shot, male and female, dog and cat. That's a pretty high bar. But the nice thing is that he funds grants, and we do fund grants that may only be female 5-year, for example, as the goal. But he's willing to give money for that kind of research because he understands that this is a very high bar. So he will grant money for things that don't quite meet this criteria, but if you want to win the $25 million, this is what you have to do. Research disciplines, we're attracting grantees from all over the world who are human cancer biologists, people who are involved in bioengineering, medicinal chemists, people who are doing the world-class breakthrough gene therapy labs have been submitting grants. So we're able to get all this expertise from human health to look at our issue. And I briefly want to point out that they're very concerned with safety and animal welfare, and there are some animal welfare guidelines that are highlighted on their website, which you can review. So the numbers. This is a slide of kitties, but it applies to dogs as well. Lots of grants were submitted. 31 proposals have been funded to date. About $12.5 million has been allocated. So I'm going to give you a little tasting menu of the different research approaches, and not too deep a dive into any of them, but just to give you an interesting idea of kind of the breadth of what's going on, and hopefully get you a little bit excited about the fact that there are some cool things coming. The new approaches for GnRH vaccines, identification of new antigens on sperm that we can target for immunocontraception, and a second look at recombinant zona pellucida proteins, which have shown to be effective for wildlife contraception, are all happening as approaches. So why a new approach for a GnRH vaccine? I already told you Gonacon, single injection, can get long-term. Well, we have this injection site reaction issue, and Gonacon is a difficult manufacturing product. So we'd like to eliminate the need for boosters, get a longer time period, and make sure we don't have the injection site reactions. So one interesting approach is making these things called VLPs, virus-like particles. So they're actually kind of artificial viruses that then get decorated with an antigen, in this case a GnRH protein. And there's this hypothesis in human health that your B cells will see something as foreign and attack it if the little decorated particles are spaced a very particular spacing on the surface of these virus-like particles. And scientists are able to make these things in the lab and make them with this exact kind of spacing. And the idea is that it will create the most robust immune response that your body can make. And a company called Cytos Biotechnology is working on this. And they're particularly interested in developing an immune response to self-antigens. And a self-antigen is one that naturally occurs in your body, like the GnRH hormone. So they're working on this technology to target contraception for dogs. There's another interesting approach that I think is really kind of jazzy, where you can put your immunogen in a gel-like matrix that you give one injection. It gives you a boost like you're getting a regular vaccine. And then it kind of goes dormant for about a month, and then it slow releases another burst, which is like a booster, but you don't have to get a second shot. And so it's kind of a self-boosting biomaterial. And this is being looked at as was one of the opportunities here, so that it's not practical for you to catch a dog twice within a month and give a booster injection. So this would be a self-boosting technology. And then people are working on viral vectors to deliver these antigens, which can incorporate into the animal's liver, for example, and produce this antigen for the lifetime of the animal. And it's really not science fiction, because some of this has actually been demonstrated to work. New immunocontraceptive targets are looking at proteins that are involved in the generation or maintenance of follicles or sperm. And there's a lot of genetic analysis being done on dog and cat, egg and sperm, which has never been funded before, but is now able to be funded through this mechanism. And one new target was recently identified at Cornell. It's called maternal antigen mater. It's an antigen that the embryos require, and they're doing a co-vaccination using the virus-like particles with GnRH and this mater to try and elicit a long-term immune response. So really unusual and new approaches to contraception that we haven't seen before. Another interesting approach is high-dose or long-term GnRH agonists. And this is a picture of deslorelin, which is the active component in Supralauren that we talked about earlier. And the idea is if you could get a slow release over 5+ years, we know this compound works. We just need to get it there at high enough doses for long enough term, and we can do that with little programmable devices that could potentially deliver this over a long period of time. We're also looking at pre-pubertal exposure. So can you give a high-dose deslorelin and keep an animal from coming into puberty so that she never has a litter of kittens or a litter of puppies? And there are some data showing that very early in an animal's life, if you give a high dose, you can significantly delay puberty. It hasn't been explored in dogs yet, but that's on the list of things to do. A really interesting approach, because again, we know the drug, we know it works. This is a new area which has been used in human cancer biology, and I won't go into all the details here, but it's taking a toxin and it's administering it in a way that it targets a specific cell. So you got to get that toxin to a cell and kill a specific cell. In some cases, the GnRH neuron in the brain, or it might be just the primordial follicles in the ovary. This is an example of that it actually works. This is a publication from prostate cancer where a targeted cytotoxin targeted with an antibody takes a toxin directly to the prostate cancer cell and kills it. And this is being developed as a human treatment for prostate cancer. So you turn this around and you look at our targets and you find a way to deliver a specific toxin to the brain, just to the neurons that make GnRH, and you can shut down reproduction for the lifetime of an animal. You can also target stem cells in the ovaries or in the testes using these homing peptides. So there's a variety of grants that are looking at this targeted delivery of cytotoxin. Again, we know it works from human medicine. We just have to apply it to our targets. And finally, briefly, I'll tell you about gene silencing and gene therapy. Gene silencing is a way of turning off a gene. Gene therapy is a way of delivering a gene. Gene silencing uses these things called small interfering RNA. They're fragments that can interfere with gene expression. They're delivered by viral vectors. And there's a grant that's looking at delivering small interfering RNA to suppress androgen receptors. There's another one to suppress maturation of ova and sperm in the testes. And these, again, are delivered with viral vectors. So shutting down important reproductive genes with technology that we know works from human medicine. And finally, gene therapy. I put this dog here because this has actually been shown to work in a dog. We've cured dogs with hemophilia by a single injection of a viral vector, which then delivers the protein that these dogs need to make them normal. And these dogs have been studied for up to 8 years and been cured of their hemophilia. So you get the right thing in there with a viral vector and you can get a long-term effect. That's just what we're looking for. So we're looking at viral vectors that deliver a variety of things that could shut down reproduction. This is a very promising area because, again, it's a single shot with a long-term effect. So now to the final question, yes, but when are we going to get these cool, exciting new tools? And, most of the research to date has been with cell cultures and rodent models, but we're accelerating the pace by all this exciting research that's happening. One thing I'd like to point out, we talked yesterday about the importance of publication. The publications from the original grants are just now starting to be published in peer-reviewed journals. These grantees are sharing information, they're communicating, they're learning basic facts about dog and cat reproduction that we never knew before. And let's face it, breakthroughs are unpredictable. But let's look back 10 years. There were no new therapies, and now we have Supralauren and we have Zuderin. There was no research or very little research on dog or cat reproduction, and now we have $50 million in grant funding. We know that when we learn about stuff that doesn't work, it actually brings us closer to things that do, because we learn so much from the failures. This is how I like to look at it. We're getting grants all the time, collaborations are forming, communication is building, and I think we really are learning what has the most potential. And I just, as a last note, would like to talk a little bit about the practical support, because once we get this toolbox of tools, we've got to have a way to really use them effectively in the field. So when we have them, we want you to be prepared and ready to use them in the most effective way. So we need to understand the challenges of how to administer these treatments. I mean, if they have to be IV, how are we going to do that? Is it possible to do that in a field situation? We have to mark the treated animals, and there was a wonderful little 2020 talk that Val Benka did yesterday about the work that ACCDC is doing to support a marking initiative. And then we have to evaluate whether or not we think these things can be effective. I mean, if we have something that's for 3 years, is that going to be effective in your situation? And population modeling is really going to help us understand that, and Dr. John Boone will be talking about that later today. So a little pitch for ACCD. We're supporting all these efforts. Our goal is to really facilitate the new stuff that's coming out and help get it out there and help you understand it and get the feedback from you about how it works in the field. And there's certainly barriers for new products. I mean, there's a lot of people who tell me, well, veterinarians just like to spay and neuter dogs. They're never going to try anything new. Big pharma is not investing. As I've talked about, there's a lot of regulatory hurdles, technical difficulties. It's very expensive to develop a product like this, but luckily we have a deep-pocketed supporter who's helping us through all this. If you want more details about many of these things I've talked about, there's an excellent ebook that's available on the ACCD website that's a deep dive into contraception, non-surgical contraception, that I'm really proud that we've put together. It's really up to date. It was brought up to date a couple of years ago, and it's a great resource. And I really do believe that we will get there. I mean, when we first started this project with the Michelson Prize and Grants group, I think we were all pretty skeptical. We've seen so much progress. It's going to be years, but we are going to get there. And I really predict that 10 years from now, we're going to be standing here and you're going to be telling me how you're using these tools in the field. We're going to get there. Thank you very much for inviting me, and I'd like to say thank you to all my colleagues who helped to put together this talk. And do we have time for questions? Happy to take any questions. I know it's a lot. That's the way up here.
The long route to the microphone.
Yes, indeed.
Thank you, Linda. That was a really great overview. I'm Kati Loeffler from IFA. A practical question to start with. One of the things that I find myself kind of having to handle with some frequency is groups who are working in the field and who are struggling with sterilization issues, and who read about some of for example, the Dogs with No Names project, okay? And they really don't quite have the context. You know, they haven't gone through the literature and haven't heard your talk, or the ACC and have read that, or really have the training to understand that. And so they want to, they pitch wanting to study this in the field, and oh, we want to use this, and this would be so simple, and let's just give it to a lot of dogs and see, you know, something. And so I find myself then in the position of having to, well, fortunately in the position of being able to suggest things that would need to be studied, and that you couldn't just, for example, give this to a dog and then 2 years later see if the dog is producing puppies. That these potential side effects can be very, very serious and cause an enormous amount of suffering. So how frequently you would need to monitor the dog, what kinds of things would need to be looked at, and those kinds of, and how to do that, and just doing a phone questionnaire with an owner might not really be appropriate, and those kinds of things. So I'm wondering what you would feel about, as the scientific advisor or for ACCD, to perhaps put together a simple research guideline, you know, for the use of these kinds of things, off-label use, and then if you would want to try it, and do a little study of your own, these are the specific things that we would need to understand, and these are key things to kind of monitor. You see what I mean?
Yeah, that's a really good point. I think it really speaks to the risk-benefit context. And I know in Judah's situation, these dogs are incredibly hard to catch and treat. They're really at the periphery of the community. It's very hard to observe them. All she really is able to do is know that they have fewer litters of puppies. Often these dogs don't live for more than about 2 years or so. They really are incredibly stressed environmentally. It would be very hard for her to do the kind of monitoring that you might be able to do in a situation that I hear a lot of you folks describe, where the dogs are really seen every day and they're really more community dogs. I think the idea of putting together kind of a checklist of the kinds of things you want to look for would be extremely useful. And there's no reason why we couldn't do something like that as ACCD. I think that's a great suggestion. In a situation where you can do close monitoring, any information you have about side effects is extremely useful. These products are also being used on zoo animals, and there's quite an extensive monitoring system to keep track of side effects in zoo animals. They're used, for example, to suppress reproduction in tigers, where the zoo doesn't have capacity to deal with so many tiger cubs. And they eventually will reverse if they want to breed that animal. So we're keeping track of how long that reversal is. But it might be a very interesting thing in a field situation to see if a year's implant actually lasts a year, or does that bitch come into heat in a shorter period of time, or maybe it lasts 2 years because she is nutritionally stressed and she's not so reproductively sound. So keeping track of side effects and effectiveness in a field situation, in all of the different situations that these things might be used, would be very valuable. And those data, even though not placebo-controlled and done under a protocol, still could help inform us. The only caution that I would say is that it's easy to overinterpret those kinds of data. So if you see one dog that develops a pyometra, and say, okay, that's it, this isn't safe, then that's a problem too. So you have to understand the incidence of pyometra, for example, in that population to say, well, maybe that wasn't a drug-related effect. So there's kind of, the data are always good. How you interpret the data can be problematic and needs some oversight. Yeah, very, very good suggestion. Anybody else?
Yeah.
Yeah. Go ahead.
Hello, Nancy Brienne from the Federation of Veterinarians of Europe. Thank you very much for this very nice overview. What struck me as I saw this presentation is that many of the new developments you presented might not only be interesting in the field of dog population control, but also in the field for other animals, such as, for example, pig castration. We're very much trying to find methods to stop surgical castration of pigs. Now, knowing that the administrative burden is very high to register a product, and of course the company that wants to register it wants a profit, it might be interesting if they have products which they can use for different species. So I don't know if you're looking into that one also, that area.
Yeah, actually, there is an approved product for pig castration that is a GnRH vaccine, and it is used in the field. It was developed by Pfizer Animal Health, but it's now marketed by Zoetis. It's called Improvac, and it uses exactly this technology. But in that case, they want a short-acting GnRH vaccine. But there have been approved GnRH vaccines for cattle, both to suppress reproduction in males and also in females that are in Asteris in feedlots. So yeah, there are definitely production animal applications for these types of contraceptives, although generally there's not the same kind of need for a long-acting contraceptive.
Well, I know for Improvac, one of the problems is that you have to give it twice or 3 times. So there also, if you could generate one that gives an automatically booster, I think that would be a big step forward.
Yeah, exactly. Yeah, very good point. Andrew?
Thanks, Linda. I'd like to talk about a different social problem that I have to deal with in my role as Chief Scientific Officer of the Humane Society. You get one of these new agents coming on the market, and suddenly everybody is saying, oh, this is the best thing, this is fantastic, when are we going to sterilize all 300 million dogs? And this happens with Nudisol. Well, it happens with Nudisol. I mean, Nudisol started in 1972 in the University of Missouri, and Lloyd Faulkner was heavily involved in those early days. It didn't get licensed by the FDA until 2003-4, or thereabouts.
2004, yeah.
It was marketed. The marketing was a complete disaster, and it was withdrawn and then licensed to Arc Sciences, who are now bringing it out as Zuterin Asterisol. It's not cheap. I mean, it costs us $12 to sterilize a dog in Costa Rica. And if you look on the website for Zuterin in the United States, you can see prices up to $60. So it's, but people sort of say, oh, fantastic, we've got this technology, let's use it. Why haven't you stopped doing all this antiquated surgery? Why aren't you sort of going to the new technologies? And so there's a huge hype factor. And I find myself then sitting in the situation of saying, yes, but, it's not going to be cheaper than surgery. It's not going to do this. It's not going to do that. It's not going to do the other thing. And it's an unenviable position in which to be, because more often than not, the people who are saying this are either board members or big donors. And so you're sort of discouraging people from in a situation where you really don't want to discourage them.
Yeah, I think that's why I'm trying to emphasize in this talk the risk benefit. Because I think everything has risks and everything has benefits, and certainly cost has to be taken into consideration there. So, and every situation may be different. So the new sexy treatments that sound really good on paper may not really be very effective in your situation. And you folks work in such a variety of different communities. So I think you have to sit down very carefully in each community and think about how much it actually is costing you to do what you're doing, how long it's taking, what the risks are, what the side effects are, and then look at the new product and say, okay, how much will this cost? And I mean by cost fully loaded, not just the purchase price, but how much it actually costs to catch the animals, to administer it. When you think about a trap, neuter, returns kind of situation, if you have to anesthetize the animal, do the surgery, recover the animal, and return them, that's a lot of cost and time. Whereas if you could catch an animal in the cage in the field, squeeze them up, give them an implant, mark them with our new jazzy little ear tag, and let them go, you can do a lot more animals for the same amount of logistics. So you really, for your situation, have to think about the cost of using it, the actual cost of the material, and then what the risks are. How easy is it for people to follow up? In terms of Zutrin, you really do have to have training in how to administer it. Who's administering it? Do you have the ability to train them in the way that to minimize side effects? What will happen if a dog has a problem? Who's going to follow up with it, both on the surgery side and on the Zutrin side? So in every situation, I think before you initiate, you need to sit down and have a very clear cost-benefit, risk-benefit analysis. And I think that will help in those kinds of conversations where you really look at the whole picture and not just, oh, this is new and exciting and let's use it in our situation. It takes some evaluation.
Thank you, Dr. Rhodes.
Thanks very much. Thank you for your time.